This presentation provides an overview of the IWS Workbench and a description of the integration between IWS with IDBS eWorkbook Suite and BioBook. Neural ID's Intelligent Waveform Service, or IWS, is a data reduction and analysis suite designed to accelerate life science research and development. R&D teams use IWS to convert large, raw signal datasets into information suitable for lab notebooks, reports, filings, and further analysis. From discovery through preclinical, clinical, and consumer, Neural ID provides the only collaborative biosignal analytics platform spanning the pharmaceutical life cycle. IWS is a scalable enterprise solution that addresses the industry's productivity crisis. IWS outputs analytics into a standard database. IWS is delivered with a suite of tools and SQL code to generate reports in Excel, IDBS BioBook, and Spotfire. Researchers can now collaborate to train IWS to recognize and measure biosignals via a user-friendly Workbench client. The consolidated knowledge base, stored with analysis templates, becomes a reusable asset. IWS can read and process data in multiple vendor formats, enabling users to manage data from discovery assays through the preclinical and clinical trials to consumer applications. Neural ID's patented Cure technology processes data to produce numerical analytics. IWS processes in minutes what would have taken days before and helps the researcher make better decisions faster. Customer studies have shown a 5 to 10x increase in productivity with a 3 to 5% improvement in the quality of results. IWS is architected for the enterprise. Key elements of the architecture are the Workbench, which supports a wide range of industry-standard data formats. Reusable and shareable tools, which promote rapid development and analysis. The job management tools provide an instant view of analysis progress, in addition to automatic notification via text messaging or email. IWS uses a client-server model where the deployments can be scaled from a single user to multi-users to multi-servers and across the entire enterprise. Oracle, SQL Server, and Postgres are some of the industry-standard databases IWS supports. Reporting tools enable visibility into the analysis with standard spreadsheet tools like Excel or advanced dashboards like IDBS eWorkbook or TIBCO Spotfire. This next part of the presentation reviews the integration of IWS with IDBS eWorkbook Suite and BioBook. IWS is architected as a client and server, which must be first connected together by selecting the Connect button. We'll start off by selecting a multi-electrode array or MEA template. IWS allows analysis templates to be built and then run as jobs against previously recorded data. Templates are reusable entities that allow you to perform analysis on waveforms in order for the raw data to be converted into useful information. The objects defined in IWS are markers, shown by the blue lines, and patterns, shown by the red boxes. These are detected in waveforms, turning the signal data into useful analytic data, which is then stored in a standard database for reporting. Once an analysis template is configured, it may be run as a job. To submit the template as a job, select the template, right-click, and select Run Job. Point the template at a source of data. We'll also add some detail to the job name, and click Run Job. Jobs are managed in the job tree shown in the lower left. In this example, the job number is 34. You can also display statistics for the current and previous jobs. Also displayed is the progress of the job itself. Switching to the IDBS eWorkbook, Neural ID, in collaboration with IDBS, has developed a BioBook add-in for IWS, which allows users of both technologies to directly import IWS job results into BioBook, as well as the ability to jump directly from IDBS back into IWS via a launcher link. You'll notice that we already have several experiments defined. We'll select the MEA experiment for terfenadine, and then we'll also select the terfenadine MEA experiment tab. In addition to the unpopulated tables and graphs where the results will be placed, materials relevant to the experiment can be added on this page ahead of time, such as the compound's molecular diagram, methods, input data files, and any other useful information, such as posters, papers, and safety notices. To populate these empty graphs with the latest results from IWS, we start the IDBS BioBook software, and here we see tables that are pre-configured to receive the results. In this particular example, we're going to perform an RR interval report and a QT interval report. Then, from those, we're going to calculate various corrected QTs, including Bazet, Fredericia, and regression. To import the IWS database results without any transcription errors, we'll just use the data link. We'll edit the data link for this table, select the IWS data link, connect to the database, select the job, which can be identified by either its number or its name, and finally we select the type of report to be generated, in this case an RR interval report. Clicking on Next gives a preview look at the table, and we'll select a two-dimensional table and then Finish. We can see the progress of the fresh data being imported. Similarly, we bring in the QT interval data by establishing and editing the data link, connecting to the database, selecting the same job, and then selecting the QT report. Clicking on Next gives the preview look at the table, and we'll again select the two-dimensional table and Finish. The data from the latest job has been brought in. We can see that all of the tables have been populated. The charts have also been brought in. By saving the sheet and exiting BioBook, we'll be returned to our experiment page. Notice that the charts have now been populated with the IWS results for the various concentrations. We can see that terfenadine is prolonging the QT interval as shown by a definite stair-stepping on two of the electrodes in the multi-electrode array. In addition, we can display the automatically calculated corrected QT interval values, which also demonstrate QT prolongation. Now we'll demonstrate another experiment which will use preclinical data and will show the full capability of BioBook's Click Back feature. Entering BioBook, we see that there is already data available generated from a previous job. As we look through the time occurrences for this channel and the QT interval, notice that one of the QT intervals is missing. The Workbench Launcher allows you to double-click on the launcher link, which will automatically launch IWS into the analysis template and take you to the exact point in time the pattern occurred. Back in the IWS Workbench, we see the reason the pattern was missed. Instead of being a standard QRS, there is a type of arrhythmia present, referred to as a ventricular complex. To explore this arrhythmia a little further, we can quickly modify this template to detect all the ventricular complexes in the data. To make the display less cluttered, we'll disable the T pattern detection. To create a new pattern, we'll zoom in on the region and then we'll add a new pattern to the analysis template by drawing a box around the ventricular complex. We'll label the new pattern VC for ventricular complex and link it to the original right marker because the marker is just a little right of the pattern's leading edge. And we'll increase the detection tolerance so that IWS will accommodate some variation in the shape of the VC. Now that the VC pattern is defined, all that remains is to learn the pattern, which we'll do with a right-click and selecting Learn. We'll do a local analysis to verify that the pattern is being properly detected. Now we'll save the template and exit setup mode. The preclinical template will now be submitted as a job. We'll use the default settings and select Run. We can follow the progress as the job moves through the job tree. There were 166 ventricular complexes detected on job 35. We'll close the IWS workbench and switch back over to IDBS BioBook. You can see that we already have a table established for pattern detections. A data link must be established again. We'll edit the data link, connect to the database, select Job 35, Preclinical Ponema VC Detection, and then select the Detected Patterns report. The table preview will be displayed. All of the VCs are displayed with their relative timestamp. A two-dimensional table will be selected, and the results data will be imported. The Detected Pattern report is a chart that's already defined within BioBook, and which displays the occurrence frequency per minute of the ventricular complexes. The chart is automatically generated because we've previously defined a table containing the necessary calculations which use the imported data. This chart now becomes part of the BioBook report in eWorkbook. The workbook can now display sheets containing multiple charts and tables, such as the time bin chart. The charts and tables are visible at the top level, which now display the frequency occurrence of the ventricular complexes. Going back into BioBook, where all the ventricular complexes are listed, we may want to explore deeper into the experiment. We simply double-click on the Workbench Launcher link at the location to be examined. The IWS Workbench will be reopened and the timeline will be positioned to show us exactly the occurrence of that particular ventricular complex. This video has demonstrated the tight integration between NeuralID's Intelligent Waveform Service with IDBS's eWorkbook Suite and BioBook. Thank you for watching this instructional video, and we hope it was beneficial. Contact NeuralID or IDBS for more information or for a hands-on demonstration of these systems.